So we are talking about blood. We're already to chapter 20. So good stuff. We're getting into the cardiovascular system. And the cardiovascular system is a system because it is the heart. And the heart is a muscular pump. It has one job, to contract as a muscular unit, and its job is to pump blood. So the contraction causes the pressure that pushes the blood through this closed circulatory system. That is the arteries and veins that we name in the circulatory system. That's chapter 22. And what the heart is pumping and goes through the circulatory system is blood. And so blood is like the transportation system in your body. So diffusion only works in short distances, but the cardiovascular system can transport things in your blood long distances. So blood is classified as fluid connective tissue. It's specialized connective tissue in which it distributes nutrients, oxygen and carbon dioxide are gases, and also distributes hormones that we talked about in chapter 19. Can also take away the waste products. And the kidneys, I don't know if you've realized, but the kidneys are responsible for filtering the blood. So the kidneys filter waste out of blood. Let's see if I can write that about it. Filters waste out of blood. And it still looks pretty sad, right? Filters the waste out of blood. So the blood gets filtered by the kidneys. There's also white blood cells. White blood cells defend against disease and infection, and also the transportation and regulation and protection. Okay, let's talk about transportation. It's not just nutrients, oxygen, carbon dioxide, hormones. You have ions, right? So sodium, calcium, potassium, magnesium, so forth. So you have all these ions, right, that get transported. You have hormones that get transported. You have lipoproteins that get transported. So there's a lot of stuff. You have fibrinogen and platelets. So a lot of stuff's getting transported. Whole blood is just what you see kind of pouring out of your body when you see your blood. But it really can be broken down into two distinct components. So the first component is plasma. That is the liquid portion of the blood, and there are dissolved proteins in the liquid portion. That is where the nutrients are, the ions, glucose, dissolved gases like carbon dioxide, and hormones, so forth. The second part is called the formed elements, and the formed elements are really three distinct things: red blood cells, white blood cells, and platelets. So red blood cells, white blood cells, and platelets make up the formed elements. And we'll get back to that in a minute. But again, functions, delivery of oxygen and taking away carbon dioxide, distributing nutrients, ions, hormones, and so forth. Transporting the metabolic waste away from the cells, and having the kidneys then filter out the waste and excrete it. Stabilization of pH and electrolytes. So electrolytes, again, are going to be those ions, and we're going to talk about that in physiology as well as pH. So we kind of get more to this in physio. Prevention of fluid losses by clotting. So platelets, which is part of the formed elements, number 3 of the formed elements, works with fibrinogen, which is created by the liver, to form clots, right? So platelets put fibrinogen, form clots. And when you have a break in the capillary wall, obviously blood's going to leak out because it's no longer a closed system. And so you have to close back up that system by forming a plug or a blood clot until you can repair the endothelium, it's epithelium, so it will repair itself and then go back to being a closed system. Toxins, drugs, pathogens, these are going to be white blood cells. And then keep a stable body temperature. Again, you can distribute the heat in your blood, the heat that's being produced by that inefficient transfer of energy in the skeletal muscle system. So we're transferring, the skeletal muscles are transferring energy from glucose and lipids to ATP, and then taking that potential energy that's in ATP and transferring it to make something move in kinetic energy, like the myosin heads move. Again, it's not 100% energy transfer. Some of that energy is always lost in the form of heat. And so we have this heat being produced, and the heat gets distributed in your bloodstream. You could bring it out to your skin to release the heat. That's why your skin turns red when you exercise. You have more blood flow to the skin to get rid of that heat. Or if your skin turns blue, right? So this is red, blue, then you have a decrease in blood flow to skin, right? Because you're trying to keep heat in body. You don't want to get rid of it. So the blood is what's carrying this heat. All right, back to whole blood. So this is a sample of whole blood, right? Everyone loves having that happen. And then what they do is they put this tube into something called a centrifuge. And a centrifuge is going to spin the blood really fast in a circular motion. So it spins it in a circle really, really fast, and the test tube is allowed to kind of swing out so so it goes pretty much transverse. So if this is the test tube, when it's getting spun really, really fast in the centrifuge, it swings out, and so this is the bottom of the test tube, right? So this right here swings out, and this would be the top. And what happens is when it's spinning really, really fast in the centrifuge, all of the heavier elements are going to go to the bottom, and the lighter things stay at the top. And so it separates the plasma from the formed elements. So formed elements are heavier, and they go to the bottom of the test tube. And the plasma or liquid portion with dissolved ions, solutes, plasma proteins stay at the top. And so it gets what we call spinning of blood, or you spin the blood. When someone orders a CBC, the CBC stands for a complete blood count, they're actually counting and looking at the percentage, how many platelets you have, how many white blood cells, and exactly what type of blood cells, and what their percentages are. Right, so there's 5 types. There's neutrophils, eosinophils, monocytes, lymphocytes, and basophils, and then percentage of red blood cells, right? So you're checking for O2 carrying capacity with number of red blood cells. So CBC is looking at platelets, white blood cells, and red blood cells, or otherwise known as the formed elements. The plasma portion, you can look at plasma proteins. These plasma proteins, especially albumins, are responsible for osmotic pressure or osmosis, right? You need these plasma proteins to have in your blood to keep fluid coming back into your bloodstream. So super important. Globulins transport things like hormones and lipids. Lipids are nonpolar, and they can't just cruise around in a polar aqueous environment. So they have to have these globular proteins to help them kind of survive in this polar atmosphere if they're nonpolar. Fibrinogen is necessary. It needs to get together with platelets to form a clot, proper clot. And then regulatory proteins like enzymes, hormones are in there. In addition, so those are the proteins. In addition, you have, again, electrolytes It's giving you a list, including bicarbonate and chloride and so forth. And then organic nutrients. It gives you a list, amino acids, fatty acids, glucose, so forth. And it also carries the organic waste that are going to be excreted in the kidneys, uric acid, creatine, bilirubin, things that you don't need any longer, want to get rid of. Okay, so physical characteristics of blood, it's very thick. We call that being viscous. Viscous means sticky and pours really slow or slow to flow. Obviously, the bigger you are, the more volume of blood you have in your body. And if you fall below these, you are hypovolemic. If you fall within this range, you're normal volemic, or hypervolemic would be too much. Blood is slightly basic. And when I say that, we have the pH scale for acidity, right? So 0 is 100% acid. 7 is completely neutral. And then 14 is 100% basic. So when it falls between 7.35 to 7.45, it's slightly basic. Realize that pH is logarithmic, so even 0.1 is equal to 1,000 times increase. So going from 7.35 to 7.25 is a huge drop, like deadly. The temperature of our blood is 100.4. That's why we don't really assume people have a fever unless they actually have a temperature above 100.4. Okay, so composition of blood. Let's talk about plasma proteins. It's 46 to 60% of what is in your plasma. Big part, most of it is albumins. Again, this is really has to do with osmosis, right? So osmosis. Globulins, fibrinogen, regulatory proteins. And so again, it goes through each one of these, for clots, and then globulins, antibodies or transport globulins. 55% of the volume of whole blood. I mean, it's this is your plasma, and of that, 92% of it is water and only 7% are proteins. Of that 7%, by the way, 60% of the 7% are albumins. So these percentages, I don't want you to get misled there. Differences between plasma and the interstitial fluid. That is the fluid that surrounds cells in tissue beds. That's what interstitial fluid is. Obviously is going to be the concentration of oxygen and carbon dioxide. I should say partial pressure, right? So when we talk about gases, we talk in partial pressures. And so the blood is always going to have a higher oxygen partial pressure than an interstitial fluid. So it's going to go from a high to a low. And the carbon dioxide in the plasma is always going to be lower than the interstitial fluid. So the carbon dioxide then goes into the plasma. Plasma has plasma proteins. Interstitial fluid should have 0 plasma proteins because if those plasma proteins, remember, it's creating an osmotic pressure. So say you have a capillary. This is capillary. And you have proteins in here. And proteins are solutes, right? So proteins create it's going to create this osmotic or onconic pressure, right? So it's going to create a pressure to pull fluid or keep fluid in here, right? In capillary beds, you're going to have, you need to move things out of the capillary into the interstitial fluid. So this is interstitial fluid here around cells. You're going to need things to move out, right? So you want fluid to leave and go to the interstitial fluid. So you want ions and glucose and oxygen and all that to leave and go into the interstitial fluid. But as the blood is flowing, right? So blood's flowing in the capillary this way. At some point, you got to get the waste products back into the capillary, right? So as fluid's leaving, at this end, you start having a high osmotic pressure because there's lower fluid, right? Lower fluid because fluid left at this end. So the fluid decreases, but you still have these plasma proteins stuck in there, and that's going to increase the osmotic pressure. You have more proteins and less fluid. So it creates this osmotic pressure to then draw fluid back into the venous end of a capillary. And so, so you get most, 85, 87% of the fluid back to go back to the heart. Now, if you break that blood vessel, say you get a bruise, you get hit really hard, blood vessel now opens up. And so this capillary opens up. I didn't do that very well. Let's do this. Capillary breaks. There's my break in the capillary. And blood leaves with plasma proteins. And now plasma proteins are in the interstitial environment, right? So interstitial fluid now has plasma proteins. Well, you can mend this, right? You form with platelets and fibrinogen, you form a plug, it goes back to normal, and you start having blood flow again. But you still have plasma proteins out here. So you have now decreased osmotic pressure. And so you have less osmotic pressure for fluid to come back into the cell. And so that is why you have swelling when you get a bruise. Because the bruising is the capillary walls breaking and blood flowing out into your interstitial environment or the environment surrounding your cells, right? So you can see that there's blood out there, it's purpley. And then those plasma proteins are also out there. And so it's swollen, and it takes a while for that swelling to go down because you have to really get rid of these plasma proteins slowly by slowly in order to reabsorb that fluid back into the circulatory system. So it stays swollen for a while. That's a long way of explaining to you why you should only have plasma proteins in your blood and not in your interstitial fluid. And we will talk about that again when we get to physiology. So the formed elements, white blood cells, they're also called leukocytes. That's where we get leukemia. Leukemia is a cancer of the blood cells, the white blood cells. So you can have different types of leukemia. So you have 5 types of white blood cells. And each one of these performs a different type of immune response. And so when we look at whole blood, formed elements make up about 45% of the total of whole blood. 55% is going to be the plasma. Of that 45%, 99% of it are red blood cells or erythrocytes. 0.1% are going to be white blood cells, and 0.1% are going to be platelets, right? So most of the formed elements are red blood cells, clearly at 99.9%. But let's look at the leukocytes or white blood cells. Oh, no, we're going to look at red blood cells first, or erythrocytes. So when someone checks a hematocrit level, hematocrit levels are checked when someone draws your blood and they're getting a CBC and they want to see if you're anemic and have enough oxygen carrying capacity. And so a hematocrit is looking at your packed cell volume, PCV, or also it's called the volume of packed red blood cells. It's looking at your percentage of red blood cells, basically. So the definition of hematocrit is the percentage of whole blood occupied by the formed elements. And if the formed elements are 99.9% red blood cells, then it really translates to how much of your whole blood is formed elements, which is going to be equal to how many red blood cells you have. So a normal hematocrit is 40 to 54 in males and 37 to 47. If you fall below this, you start being termed anemic. And anemic is just defined as having a low oxygen carrying capacity, or you just have a low volume of red blood cells. So that's why they check hematocrit. In the doctor's office, you can check hemoglobin. It's checked slightly different. Hemoglobin's where they prick your finger and they take out a little bit of blood, put it on a little vial, and put it in the machine. And that machine then dissolves the hemoglobin molecules, and inside each hemoglobin molecule is iron, and that iron looks green. And so this machine will shine a light through the blood, check the density of that green color, and come up with a grams per 100 milliliters of blood. So how many hemoglobin molecules in grams do you have in a certain blood volume? And hemoglobin is what carries oxygen, so it's looking at your oxygen carrying capacity. So hemoglobin looks at the same thing that hematocrit does, just in a different way. Hematocrit is looking at what is the percentage of formed elements versus total blood sample. Right, so it's a percentage. And this is just looking at how many hemoglobin molecules do you have. Both are indications of how much oxygen you can carry. So red blood cells, these are all the same. This is on a light microscope, and this is on an electron microscope. It's showing you both red blood cells. Red blood cells kind of look like those Smartie candies. They're biconcave. Right, so they're biconcaved in shape, and that means they kind of dip down here and dip down there. They're very flexible. This biconcave kind of increases the surface area because you want a lot of surface area, because you want to diffuse oxygen into the red blood cell quick. And so the larger the surface area, the quicker you can diffuse. You just have more surface area for diffusion to occur. Red blood cells have one job and one job alone, and that is to carry oxygen and carbon dioxide, so carrying gases. So this job to carry gases, its primary purpose and really only purpose is super important. And because of that, the red blood cells have developed to have no mitochondria, no nuclei, nothing else going on. So there's no confusion as to what its job is. It's carrying gases. It has hemoglobin molecules that bind oxygen and carbon dioxide and carry gases. But as a result, you don't really have much of an infrastructure, and so the lifespan of a red blood cell is not very long compared to white blood cells or other cells. So 120 days would be a long life for a red blood cell. When red blood cells have to go through capillaries, they have to kind of stack up like plates, and those are called rouleaus. So they can kind of fit through small, tiny little capillaries. So most of the proteins are hemoglobin in these red blood cells, and hemoglobin is what binds oxygen. So you get to talk about that more in physiology. So going through, the significance of a lack of nucleus, there's more room, there's nothing else being produced in those red blood cells, but again, there's no infrastructure for repair. So that can be a problem. No mitochondria. Without mitochondria, oxygen can be transported to tissues instead of being used, right? Because if you have mitochondria uses oxygen to be the final electron acceptor in the electron transport chain for making ATP. If you don't have mitochondria, you're not making ATP, and so you're not going to use the oxygen. So these hemoglobin molecules are these tertiary protein structures. There's 4 chains. There's 4 hemoglobin there's 4 hemes per hemoglobin. So each heme can bind an oxygen molecule. So you can bind 4 oxygen molecules with 4 heme. And that's what this is supposed to be showing you here. Anemia, the definition is you basically have a low oxygen carrying capacity. And there are different reasons for anemia, but again, hematocrit is what checks for the percentage of red blood cells in whole blood. And hemoglobin checks for really the amount of hemoglobin molecules, which equals how much O2 you can carry. They're both checking the same thing. So there are different types. The most common is iron deficient anemia. That is where you don't have iron molecules to make a heme molecule. You have to have iron. You get iron through your diet. And if you don't get enough iron in your diet, you're what is called iron deficient anemia. And so again, definition of anemia, body has low oxygen carrying capacity. If you can't carry oxygen to your tissues, you cannot. So if you have low O2, then you have low ability to produce ATP, and you have low energy, and cells struggle, right? So you need oxygen, right? Other types of anemia, by the way, there's pernicious anemia. So to make to make red blood cells and hemoglobin molecules, you need iron, right? So you need iron in your diet. You also need vitamin B12, and you need amino acids, because those are the building blocks of these protein chains. Now, vitamin B12 is hormonally absorbed into body from small intestines, right? So from food. And if you don't have enough vitamin B12, you also can't form proper hemoglobin molecules. So pernicious anemia is where you don't have enough vitamin B12. Iron deficient anemia is when you don't have enough iron in your diet. You can also have altitude anemia. So, right, altitude, as you go up in altitude, you have a lower percentage of O2 in the air. And if you have less oxygen available, you're going to have to then increase number of red blood cells to capture less O2 available. So what happens in altitude anemia is you go up in altitude, there's less oxygen in the air, and so you actually end up with a lower partial pressure of oxygen in your blood, and you feel tired. This will then cause kidneys to release erythropoietin. And erythropoietin then stimulates in bone to produce more red blood cells. And when you increase red blood cells, you increase O2 carrying capacity. And then you end up back at normal partial pressure of oxygen. This is why people train at altitude. When you train at altitude, so training at high altitude will increase numbers of red blood cells, which will increase O2 delivery to working cells. And if you have more oxygen being delivered to working cells, then you increase ATP production. You have more energy than competitor, right? So you want to have a high oxygen carrying capacity because it's one of the limiting factors when you're competing, especially in endurance events like cycling. So like Lance Armstrong, he not only injected synthetic erythropoietin called EPO, but he also did blood doping, where you take out pints of your own blood, your body replaces it, and then you add in that extra blood, because the more blood you have and the more red blood cells you have in that blood, the more oxygen carrying capacity you're going to have, and more oxygen is going to equate to more energy production to keep your working muscles going. All right, blood type. So on the cell membranes of these red blood cells, you have what are called surface antigens, or you have no surface antigens. So type A is going to have a specific surface antigen called A. So we're going to call them little circles. And if you have type B, then you don't have circles, you have triangle surface antigens. We just call those surface antigen B. Now, type A surface antigen red blood cells have antibodies against B surface antigens. And type B have antibodies against A surface antigens. That is to say, if you give someone with type B, type A blood, they have antibodies that are going to attack those cells and cause problems. So that's why you can't transfuse someone with a different type blood, unless you have type O, which clearly has no surface antigens. And so you have no surface antigens, there's really nothing to attack, right? But you also have antibodies against surface antigen A and B, so you can't give type O, right? That would be bad. And that would be bad because you have antibodies that attack both of those, right? So we call type O the universal donor. So type O is universal donor because no surface antigens. And so you can donate them, even if they have antibodies, there's still nothing to attack on the surface or membrane of that red blood cell. The universal recipient is type AB, because yes, they have both surface antigens, but they don't have any antibodies. And so they have nothing to attack. So they're kind of like, well, all right, you can give me A, you can give me B. You didn't even give O. There's no surface antigen. So they're the universal recipient. They check this through, you can do a simple blood test, and it goes into antiglycins. And I'm not sure if you'll go over this in lab, but we could talk about it if you're curious about how you can do like a blood type, but these slides go into it. Most people say like I'm A positive or A negative, or B positive or B negative. The positive negative refers to this surface antigen called Rh factor, and you either have it or you don't. So if you have it, you're positive. If you don't have it, you're negative. This is genetic. You're going to have red blood cells, same type as either your mother or your father, or both. So like I'm type O and my husband's type O, and all three of our kids are type O because there are no other options, unless of course, I guess I had a kid with someone else who wasn't type O and they would have another type blood, but that would break up my marriage. So I wouldn't do that. But that's why they used to check blood type to see paternity. It's not great because if everyone has type O, then chances are everyone's going to be type O, it's not as easy to trace as genetics when we're looking at DNA. So this goes into blood typing. Most people in the US are type O and type A, especially if you're Caucasian. So Caucasians tend to be predominantly O and A. Remember, it's genetic. You have generally the red blood cell type of your parents. It's different for every different type. Native South Americans, 100% type O. I'm sure if there's interracial kids, if a native South American starts mating with a Caucasian, you could have more type A's popping up in the population. So I don't know how current this chart is, but it's really just to illuminate that this is a genetic thing. And so again, it goes into how to blood type. So you guys can read through these slides. You have these PowerPoints. This is what it looks like in a lab or setting. If you give someone the anti-gluten for A and B and they have those surface antigens, it makes the red blood cells fall apart or lyse up and clump up. So clearly if it's clumping up, they have surface antigen A and they have surface antigen B. It's only clumping up with the anti-gluten A, so they have surface antigen A, but no B, right? So, so we know there's type A, right? So this would be AB, that would be type. And then same here, they don't have A, but they have B, surface antigen, so you know it's type B. If there's no surface antigen, you're not finding any A and you're not finding any B, then you're an O because there's no surface antigens on type O. White blood cells, you know, they don't comprise a lot of the packed cells or formed elements. And a low count is called lymphopenia, or lymphocytosis is too many. You do a differential count. A differential count is when they look at each one type of the 5 and give percentages or a count of how many for each one of those 5 types of white blood cells. White blood cells can have a short lifespan to a very long lifespan depending on the type of white blood cell. And so let's talk about those. Most white blood cells are circulating. Many have a short lifespan, especially the highly mobile ones like neutrophils that are really going after viruses. All 5 of them are some form of defense against invading pathogens, but they also do different types of responses. So 3 of these types of white blood cells have little granules in them, and so they're called granulocytes. That's going to be the neutrophils, the basophils, and the eosinophils. It looks like the monocytes have granules, but they don't. So they're agranular, or without granules are the monocytes and the lymphocytes. Neutrophils are the most common, as well as lymphocytes. So lymphocytes and neutrophils really account for the majority of your white blood cells. Neutrophils contain chemicals that kill bacteria. So these are generally the first on site when you have an infection. These are the ones that are phagocytotic. They have those enzymes. They engulf or phagocytose or endocytose these bacteria and viruses, and they merge it with a lysosome and they break it down. They're highly mobile, but they have a very short lifespan because they're super active. Lymphocytes are more for specific immunity, and when you have T cells, B cells, and NK cells, and we'll talk about lymphocytes more when we talk about the lymphatic system, but they're the primary cells, primary white blood cells of the lymphatic system, and they're for specific immunity, whereas like neutrophils are generally just going after everything. Eosinophils, they stain with eosin, which gives it this orange kind of stain. They're polynucleated like neutrophils. So both of these are polynucleated. So they have a lot of nuclei because they're creating enzymes, right? So a lot of enzymes. And so they have a lot of nuclei to crank those out. Eosinophils are when you have inflammation. So if you have an allergic reaction or parasitic reaction, eosinophils are going to go up. So when someone does a complete blood count and you have something that's off, and you go in and they do a CBC and they do a differential count of your leukocytes or white blood cells, and they see that your eosinophils have gone up, they're going to start looking at an allergic reaction or a parasitic infection. If your neutrophils are way up, they're just going to look at a general bacterial infection or a viral infection. If your basophils are high, basophils discharge histamine to get an inflammatory response, right? Because histamines dilate blood vessels, and they also have heparin releasing to basically prevent blood clotting. Here's the thing. If you have a lot of histamine, you have a lot of inflammation. These are usually autoimmune or allergic, right? Responses. So an increase in basophil in your differential, you would start thinking, okay, is there autoimmune issue going on? Is there lupus? I mean, what are we looking at? Monocytes, they're these large phagocytotic cells. They tend to be more fixed, and they engulf pathogens and try to disable them. So this kind of goes through this chart of all the formed elements: red blood cells, white blood cells, and platelets. Last to mention, platelets, they're these little membrane enclosed packets that are like chipped off this giant cell called a megakaryocyte. And these little packets contain enzymes and proteins to form blood clots. So a platelet has to kind of get together with fibrinogen in order to have a blood clot formed. There's a whole clotting system or cascade. It's called hemostasis. And so it's the transport of these enzyme chemicals for the clotting process with fibrinogen forming a temporary patch. And then you have active contraction of this clot. There's actually actin and myosin filaments that will pull the sides together as the epithelium is being repaired. So thrombocytopenia is when you don't have enough platelets. You can get platelet transfusions. Thrombocytosis, you have too many platelets. So there are cancers or infections or diseases that cause that. So we can have platelet disorders. And last to mention is hemopoiesis. That is blood formation. So this is in generally the long bones. You have hemopoiesis going on where you start with the hemopoietic stem cell. So this is our blood stem cells. And from that, it goes into a lymphoid stem cell or a myeloid stem cell. Now, lymphoid stem cells are going to become lymphocytes. So lymphoid stem cells really have one fate, and that's to be lymphocytes, either T cell, B cell, and NK cell. We'll talk about the differences when we get to the lymphatic system. Myeloid stem cells then can differentiate into eventually becoming neutrophils, eosinophils, monocytes, and basophils, as well as it can become a megakaryocyte and become platelets, or can become an erythrocyte, right? A matured red blood cell. So there's reticulocyte is a slightly immature red blood cell, and then an erythrocyte. So hemopoiesis is the process of this blood cell formation in the red bone marrow. And these cells also, like I said in the beginning, the kidneys can secrete erythropoietin, but you also need amino acids, iron, and vitamin B12 to actually cause this to occur, right? To increase red blood cells if EPO is released by the kidneys, right? So you have to have all three of those things in order to have healthy red blood cells. And that's going to complete blood. Our next lecture is going to be the heart, which is chapter 21, and we'll finish up the cardiovascular system with the circulatory system, talking about arteries, veins, and capillaries.